In the Philippines, cancer is considered as the third leading cause of mortality. It is a disease that does not favor any gender, age, and social positions. Chemical and radiation therapy are dreadful procedures that affect not only the physiological and emotional states of the affected individuals and their families, but are also costly.
One of the critical events on the metastasis of cancer is angiogenesis, which is the formation of blood vessels from pre-existing ones. Moreover, tumor growth and metastasis are known to depend on angiogenesis. With this said, targeting angiogenesis is one of the most options to arrest tumor growth and invasion into tissues. In addition, it was also thought that anti-angiogenesis drugs prevent the growth of cancer cells by blocking the development of new blood vessels.
Plants and plant materials have been used to produce effective and cheap drugs, and some plants and plant extracts have been investigated and proven to exert anti-tumor or anti-cancer properties. With this taken into consideration, we decided to test and highlight the proven research-related botanical and pharmacological medicinal properties of aloe vera, a plant in which many researchers use as a subject to study.
The chemistry of the aloe vera has revealed the presence of more than 200 different biologically active substances, which include antimicrobial, antibacterial, antifungal, and antiviral activities of the non-volatile constituents of the leaf gel.
This motivated us in conducting the study entitled The Effect of Aloe Vera Leaf Ethanolic Extract Against Angiogenesis.
200 grams of aloe vera leaves were gathered, secured on a newspaper to prevent loss of water, and were chopped and then submerged with 95% of ethanol. The plant sample was macerated for 72 hours at room temperature and was filtered to separate the solid residue from the supernatant.
The different treatments were prepared using the formula percent by mass of aloe vera leaf extract is equal to mass of aloe vera extract multiplied to 100 over the mass of solution that led to the following concentration. For treatment 1, 2, 3, and 4, the concentration of the aloe vera leaf extract is 25%, 50%, 75%, and 100% respectively that were compared to the positive and negative control group.
18 filter papers were punched with 2-hole puncher to form paper disc and were sterilized by autoclaving, then were placed on each cell of a titer plate. These were left covered for 5 days. On the 7th day, egg membranes were upsized and then soaked filter disc was applied directly to the CAM assay. The treated eggs were sealed and were kept in an incubator for another 3 days. Through the help of a licensed veterinarian, the blood vessels branch points at each area of the quadrant was counted manually. The CAM vascularity inhibition percentage was calculated using the formula number of untreated branch points minus the number of treated branch points multiplied to 100 over the number of untreated branch points.
Phytochemical analysis showed the presence of secondary metabolites such as alkaloid, flavonoid, saponin, steroid, tannin, and terpenoid. It can be remembered in the study of Raphael 2012 about phytochemical analysis on the secondary metabolites of aloe vera leaves ethanolic extract using maceration method for 3 days that it detected the presence of alkaloids, flavonoids, steroids, terpenoids, glycosides, and carbohydrates.
These metabolites possess anti-cancer potential activities as they inhibit tumor angiogenesis and growth via blocking signaling the transducer and transcription of cell proliferation.
This table shows the mean CAM vascularity inhibition percentage of different treatments in CAM assay. It could be gleaned from the table that treatment 4 got the highest mean in terms of CAM vascularity inhibition percentage.
There is a significant difference between and among the treatments, however.
There is no significant difference between treatment 4, 100% aloe vera ethanolic extract, and treatment 5, which is the celecoxib. This implies that treatment 4 can be an alternative to the commercial anti-angiogenic drug celecoxib.
This was probably because of the chemical constituents showed in the phytochemical screening. It was found that in aloe vera ethanolic leaf extract, it contains tannins, alkaloids, terpenoids, sterols, and flavonoids.
Thus, the researchers recommend that anti-angiogenic medicine formulators should also try the potential of aloe vera leaf, and further studies should also be conducted comparing aloe vera leaf with other available commercial anti-angiogenic drugs.
We create innovations for the future generations.